Yeah, that's how we roll here.
That's great. Good morning, and thanks for inviting me to join you and share with you some of our work. Let me start by saying I'm a basic scientist, and I tend to emphasize data as opposed to overall concepts, but I'll try to do a bit of both of them today. I've been a pain researcher for much longer than most of you, and my interests are, for the last 20-something years, has been specifically to understand what are the mechanisms of chronic pain in general. So I cannot cover all of the work we have done. I'll sort of give you an overview, and if I'm losing you, please stop me and ask questions. This is an informal setting, and I would like it to be like that. So overall, what I would like to talk to you about is literally the mechanisms of chronic pain. And I will start with some basic definitions and then go over some of the data that we have. Again, please interrupt me if I'm being too obtuse. So instead of starting with pain, I want to start with what is vision. We all agree that there are some red apples and some green apples on the screen, right? There is no controversy. On the other hand, veridically, in the actual outside world, there is no such thing. This is something that our brain has created, right? Our brain has created by specific pigments that we have that filter the outside electromagnetic field, which is a fuzzy field overall, but we actually make the machinery biologically to, in fact, see this, and we all agree that we're seeing it. So, yes, these wavelengths exist outside of our visual system, but these colors do not exist outside of our subjectivity. So what? In comparison to that, what is the difference between pain versus vision? And, you're all sitting on your chairs. Everybody is putting a force of about, let's say, 60 kilograms on their chairs. But right now, at least, no one is in pain, right? Now, you may start feeling pain as I keep talking, but all of you, as you're sitting down, you're also fidgeting on and off, maybe because of what I said or not, but by and large, you are fidgeting to protect your body from feeling pain. So you have a 60 kilogram of weight on your behind that doesn't feel like anything, right? You don't feel your weight. So subjectivity also steals away information. You don't want to know your weight at any given moment. On the other hand, that weight is definitely large enough, the force is large enough to activate nociceptors, but those nociceptors do not give rise to pain. So nociception and pain are not the same thing, and pain develops over time. That chair will become painful if you sit there and don't fidget, let's say, for the next hour, right? So, in a sense, let me see. Yes. So overall, at least we emphasize that nociception is ongoing all the time. Nociceptors are active all the time, and nociceptors are actually protecting your body from injury. Most of the time, most people don't have pain. They don't have pain because their nociceptors are protecting their bodies from injury. The outside environment is noxious and is debilitating to the organism. The only way to survive it is to have nociceptors. So, on the other hand, at least acute pain is a conscious state of specifically a failure to protect the body from injury, in a sense that it's a signal that something has happened, that my standard nociceptive protection of the body is not enough anymore, and I need to either scream, write poetry, and or most often do a coordinated body response to move away from that painful situation. So and within that spectrum, chronic pain, the standard definition is pain that persists after healing process. From my behavioral sort of viewpoint of pain as a coordinated behavioral expression with subjectivity, then chronic pain becomes pathological simply because there's no more behavioral repertoire with which you can compensate for the pain. So it's a different take on the definition, but it's one that I think, at least given our research, it makes more sense. So I'm giving you these definitions as subtle differences from the standard clinical or the IASP definition of pain. And in a sense, this is what we would simplify and make all our scientific data much more coherent. You all know that the chronic pain is highly prevalent. It has a huge cost on society, and it is a condition for which we have no scientifically established treatment options. All treatments seem to be equivalent. They all help the patient for a third of the population, decreasing their pain by 30%. So that's just not good enough, and my contention has been for the last 20 years is that we just don't know the mechanisms to be able to develop more efficacious treatments. Okay, this is my favorite slide. This is a slide that was drawn by René Descartes back in 1644, and this is his conceptualization of what is a sensory system overall. In the drawing, is there a pointer? There is no pointer. It would be nice if I could get a pointer, actually. I can show you. Okay. Maybe I can do this. Yeah, okay, here. So, in a sense, we have a stimulus. That stimulus is going to be transduced into something here, which René Descartes did not. Thank you. Thank you. So there is a transduction process here that he's illustrating in some fuzzy way, a transmission mechanism, and a representation. In a sense, those are the fundamental components of every sensory system, and pain, as similarly with all other sensory systems, have been studying all of these components, and a lot of the molecules, the circuits, etc. But in the case of pain, over the last 20 to 30 years, what we have sort of established is that as soon as the painful environment stimulus is persisting, then there is local reorganization of transduction mechanisms, spinal cord reorganization, and we have essentially pioneered the concept that there's also reorganization in the brain as well. So then the general question becomes who and where is the egg and the chicken, and how these things are interrelated with each other, right? And that's kind of what I want to talk about. So here's my general outline. What I want to emphasize today is that acute pain and chronic pain are distinct brain states. So studying acute pain does not really give us insights about chronic pain. And we do have now a signature of a patient having chronic pain, and our signature is sensitive enough that we can, in fact, not only identify a patient being in chronic pain, we can actually, from a 10-minute brain scan, we can say how much pain that patient is suffering right now as I was scanning them. I'm going to spend more time on our longitudinal studies. Again, the main message from those studies is that the limbic brain properties, the brain regions that we think of them as the older parts of that have to do with emotional assessment, motivational behavior, and all of that. We think of those as the primary risk factors. And in fact, I will also spend some time showing you the elegant equivalence that is happening that now bridges the science of chronic pain in humans with animals. Okay. So an overview of how we think of chronic pain is this 4 boxes. We think of there being risks for chronic pain. Not every subject develops chronic pain with the same injury. Very small subgroup of patients with the same injury go on to develop chronic pain, and that must already imply a predisposition. Then there's an injury event, and the interaction between the two gives rise to this transitional state, which in turn creates the chronic pain organism, which is maintained for years or a lifetime. So what I want to emphasize here is that the traditional view is that all of these components are driven by simply primary afferent properties and spinal cord circuitry, which we at least have data that shows that's not correct, at least for some chronic pain conditions. So our overall view is that limbic emotional circuitry are the risk or the predisposition, and this emotional learning underlies the transition. So the transition state, combining the injury with predisposition, gives rise to a learning process, and an emotional learning process, which creates the chronic pain state, which is a new and distinct anatomical and functional brain. Okay, so for many years, us and many other groups have studied the cross-sectionally, basically the chronic pain population in comparison to healthy subjects, right? And this is where we have used a whole variety of methods. I'm not going to spend much time on them to look at the brain properties. I do want to show you some of this data. This is a meta-analysis toolbox that's available freely on the internet. You can just go to neuroscience and put the word pain. It looks in PubMed at all the papers published with brain imaging with pain and generates from that a brain map. So this brain map is the sum total of 224 studies of pain, and it's sort of the average of 2,000 people for pain. But the caveat is that 95% of the data is derived from acute pain studies. So if I compare this map to a map we have generated, this is more than 10 years ago, it's our first brain imaging study for chronic pain. The medial prefrontal cortex is activated in the chronic pain patients. It's a region around here or around there, which is not even in that overall group data. So either we're doing something stupidly wrong, or we have uncovered something new. And that's the excitement of science. And we've claimed that we have uncovered something new, right? Which is why I'm standing here. Anyway, similarly, we have also looked at the brain anatomy, just very simple measures of the cortical volume or cortical thickness. Again, this is the first paper that we published, I think, more than 10 years ago, showing that there are brain regions where the structural properties of the brain have changed. For the most part, we see decreases in gray matter density. This is in chronic back pain patients, and these studies have now been replicated in hundreds of other groups and studies, and I'm showing you some very old data from other labs, all of them showing some brain anatomical reorganization for different types of chronic pain, and with the suggestion that this cortical reorganization may be specific to the up to the type of chronic pain and maybe to the specific individual as well, which we have not really pursued. Oh, I'm sorry. Tension headache, fibromyalgia, irritable bowel syndrome, and this is chronic back pain, right? And then I'm making a small note here in that there are other studies, not necessarily by us, who have shown that these anatomical structures do reverse, at least in some part, in patients where there is successful partial recovery from their pain. So I want to share with you this data. This is a little more complicated, but it's the brain anatomy. Now I'm simply plotting the brain anatomy as a heat map. The details don't matter, but it just shows what brain regions are more interrelated versus not interrelated with each other. The blues are negatively correlated brain regions. The reds are highly positively correlated regions. This is in healthy subjects. Here is the same data in chronic pain patients. This is chronic back pain, CRPS, osteoarthritis. And you don't need statistics to see that this self-similarity of the brain anatomy is dramatically different in different groups, and it's different in different ways in different groups. If we actually now plot this data as at an individual subject level, and plot this overall reorganization as an overall index in each subject. So on the x-axis, I have pain duration. On the y-axis is the overall brain reorganization. And these beautiful curves gives us the time constant of the brain reorganization. So here you can see that the brain reorganization plateaus by around 11 years. This is in chronic back pain patients. It plateaus by around 2 years in CRPS patients. And in the osteoarthritis, it's somewhere in between. The data is not very nice. We cannot really model it. But the brain is undergoing continuous plasticity reorganization in the scale of years to decades. And we don't understand exactly what's going on. What is clear is that the system is continuously changing. Okay, I'm going to change topics. And this is my favorite slide that I want to share with you. This is a single subject brain activity. The brain has been flattened, so it's 1/2 of the brain. And this is 5 minutes of data. What I want to impart to you is this general concept that the brain has these massive waves propagating within it. And those waves, we can study them in what is called resting state brain, fMRI. Right? And we basically, for example, if I look at the activity here versus activity here and plot them relative to each other, I can quantify how much information is being shared across areas. And that's kind of analysis has been really fun. There are thousands of papers published with this technology. We can identify memory properties of subjects, their learning properties of the subject. We use this to, again, identify now the dynamic sort of information exchange in the brain and how that's changed in a chronic pain condition. And in fact, from that data, we can derive a global information disruption index, a single number, basically, that we think is specific to the presence of chronic pain and its intensity. I'm not going to go through the details of the analysis. Let me just sort of skip through some of this data, otherwise we'll never finish. But overall, we see that the amount of information in the brain that is being exchanged across many areas is disrupted. It's disrupted with an index. This is the amount of disruption for 3 different chronic pain conditions versus healthy controls. Not only it's disrupted in a very specific pattern. The amount of disruption, here each dot is the disruption index in every patient in the study. The amount of disruption is directly proportional in all 3 groups of pain patients to the amount of pain that they were experiencing at that moment. Okay? The more pain they have, the more the information is disrupted. So this is kind of what you guys all struggle with, right? These are patients where their information processing abilities are disrupted. They're disrupted in proportion to the amount of pain that they have at that moment. And the disruption is global. In a sense, I can look at we look at this data when we throw away 99% of the brain regions, the result is the same. So it is propagated across the whole brain. It's impacting on everything that patient is doing. And we can, in fact, this is the evidence that we can, in fact, do the opposite. We look at individual patients about how much disruption they have. From the model, from the original data, we can simply calculate how much pain they have. Okay? This is a completely unbiased validation approach where we are 60 to 70% correct in telling how much pain any given patient has by this 10-minute brain scan. Okay, again, let me change gears. And so everything I've told you so far are based on cross-sectional studies, comparing patients versus healthy subjects or groups of patients with each other. But again, there's this issue of what is the chicken and where is the egg, and the only way to untangle that is to do a longitudinal study, which we've published a number of years ago. And here's the general concept. Again, I've put this overall conceptual idea on top of it. So acute or subacute patients will be somewhere in this transitional state, right? And if we follow them over time, they turn into this maintenance state, which would be the chronification. So if we take a group of subjects at somewhere around this time, follow them over a year, now in fact we follow them over to 3 years, we can look at what's happening in the brain. So the changes that are happening in the brain, those are consequences, right? Those are what creates the condition. If we find things that are present at the time of entry into the study and are not changing, then we can look at those, we call them as risk factors or predispositions. That's a simple concept. And in a sense, these properties have to be able to predict in the future how the subjects are going to be parceled out into persisting versus recovering patient groups. So here is just the pain properties of these patients. They all started with the same amount of pain at time 0, the first time when we scanned their brains. But very quickly, they start segregating into 2 groups. This group are the recovering subjects, that is, the pain is going away. And these are the persisting. Their pain is exactly the same a year later. It has not changed at all. Okay? And that's the group difference. And it's also fun to look at all of these questionnaire outcomes in these patients. What we see is that the patients who are recovering, all these numbers are going down, and the patients who are persisting, essentially nothing is changing. All of their questionnaire outcomes are the same as their simply assessment of their pain. So, and let me show you the brain activity in relation to this. Okay, so here is, again, the recovering group versus the persisting group. This is the brain scans we have done at time 0, 7 weeks, 30 weeks, 55 weeks later. You can see that the brain activity at this time is identical between the 2 groups, and it actually matches the acute pain map I showed you in that meta-analysis. Okay? In time, the people who are recovering, their brain activity is decreasing. In fact, by 1 year, we don't see any brain activity anymore. Okay? Now, they still have some pain, but it's small and variable, so we cannot see any brain activity in relation to that. The persisting patients, on the other hand, their pain is exactly the same 1 year later. Their brain activity is continuously changing, and in fact, it ends up, again, activating the medial prefrontal cortex, bilateral amygdala, all of these emotional limbic regions of the brain, and which matches nicely with our original cross-sectional study. So in a sense, although by simple report, the patient's assessment of their pain has not changed at all. Their brain activity has completely shifted. It's shifted from here to there. That's a massive reorganization in the scale of the brain. And the shift is essentially implying to us that the pain is becoming internalized, right? So that it's not now a noxious stimulus from the outside environment, but it's something that my body is suffering from in a sense. And it is engaging massively all the emotional circuitry of the brain. Okay, let me move on. Again, I'm not going to show you all the details of the data. So specifically, within that data set, we really want to understand the transition properties. And within that system, we end up finding a very simple relationship. This is the amount of information shared between two areas, the medial frontal cortex and nucleus accumbens. And this interaction, we think of it as specifically the interaction of the injury and the predisposition. So 1 plus 2 is creating 3, the transitional state. Okay? And we think of this as a mesolimbic or emotional, motivational brain circuitry learning process. And all of this, I want to summarize as the brain becoming addicted to nociceptive inputs. Okay? Because this circuitry specifically is the brain addiction circuitry. All drugs of addiction engage this network. And so all of a sudden, chronic pain has become a central addictive circuitry engaged condition. And we have a lot of data to support this. I'll show some additional data. I just want to emphasize the general concept. In a sense, when we identify brain circuitry, that brain circuitry redefines what is chronic pain. Okay? Okay, so here is the data. I just want to show you, for example, this is in fact the amount of connectivity between these two areas at time 0. The first time they came into the lab, that same relationship holds at 7 weeks, 30 weeks, 55 weeks. It's not changing at all. Okay? So this is a prime candidate as a risk. So what it says that the risk factor has to do with the brain addictive circuitry properties per se. Again, this is not very important, but in fact, just this single number is highly predictive at both 6 weeks, 30 weeks, 1 year later, about by 80% correct. This single number tells us who 1 year later will develop chronic pain. Okay? And it's a validated outcome. Again, if I take a new cohort, only look at these numbers, I'm again 80% correct to tell you who developed chronic pain 1 year later. Okay? Now we've also extended these analyses up to 3 years, and essentially again the pain properties remain the same. There's 2 subgroups that essentially bifurcate by about 3 months are not changing at all. 3 years later, that bifurcation is the same. People who start developing signs of chronic pain will continue into chronic pain for years later. Right? Very few of them will revert back, at least in this data set. Okay, but more importantly, since I've been emphasizing the limbic brain, in this study, we actually looked at the limbic brain specifically and created a network approach to analyze that data. From that, we identify a specific network. This, in this case, is again frontal cortex, nucleus accumbens, and amygdala. The 3 together, their anatomical properties, their functional properties, both of them predict again who's going to be chronic pain patient a year or 3 years down the line. Part of that network is also anatomical properties of that region. Here, we're looking simply at the hippocampus. The hippocampus, you all know, is an important area about learning and memory itself, right? As well as emotional assessments. But simply, if we calculate the volume of the amygdala, the volume is significantly different at all time points in the patients that are persisting versus in the recovering patients. And that difference stays three years later, and it's applies to both the left and the right hippocampus. I'm not showing you the data. We see essentially the same result if we do the same analysis for the amygdala as well. So at least the anatomy of the amygdala and the hippocampus, again, are risk factors for developing chronic pain. And these are emotional learning circuitry. These are the fundamental emotional learning circuitry that drives, humans and other animals into behavior. So to summarize in a sense, again, from this conceptual diagram, I have shown you multiple shape, anatomy, and functional properties of the limbic brain, all of them predicting 1 year later who will be persisting and who will be recovering. So then we throw all of these parameters together and build a very simple model. This is a simple logistic multiple regression model. The power of the model is that we have 3 different parameters. Each one independently contributes to the outcome. The white matter properties of this limbic prefrontal amygdala accumbens circuitry, the functional properties of this circuitry. This is how much, again, information is being shared intrinsically within that network. And here I've put amygdala. I could replace that with hippocampus. The result will be the same. This is the anatomical component. All of them contribute to my model, and my model predicts the variance of the outcome, 60% of the variance of the outcome. Okay? No such data exists for any other parameter for chronic pain. There is a lot of probing and touching and pushing of skin that my colleagues have done over the last 50 years, trying to develop a model from the nociceptive sensory properties. But the best ones that are positive outcome explain at best 10% of the variance of the behavior. Here, in fact, we have taken away some parameters from the model, because otherwise my model will explain 90%, and I don't believe it. It's too good. So we want to be conservative, and this is our conservative assessment, specifically for this population. This is back pain patients, and we're looking at their specific properties. Anyway, so we use this model in fact. More recent studies, this model we apply it to every patient that comes into our lab, and we put them in the model as who's going to become a chronic pain patient versus not. So that's my overview about the human data. I want to show you a few slides from animal models. In a sense, I'm a neuroscientist, and neuroscience nowadays is in its golden age. It's really exciting. We have amazing tools with which we can look at causal relationships between neural circuitry, neuronal activity, and behavior. And we've been massively involved in this, but many other labs have helped us develop this data. But what I want to show you is that in the rats, just as in humans, there is a functional reorganization of the limbic brain. That's a time-dependent event that happens after the injury. And this brain region that we know has to do with addictive behavior, and its properties are also changing in rodents. And so we have a very nice parallel between humans and animals that we can go back and forth. Okay, so this is the global information sharing in rats. These rats have been injured by a peripheral nerve injury. This is a standard model for creating a neuropathic pain condition. These animals have pain-like behavior for the rest of their lives. But that pain is already massively exhibited 5 days later, and it becomes even worse 28 days later. Okay? So the pain behavior is already there at 5 days. 28 days later, it's again there. What I want to show you is that if I, again, do this disruption index analysis at 5 days, there is no change. So this index in the acute phase of the transition from injury to a chronic state does not show up. But 28 days later, the same animals, right, they all have very similar pains. Now I see a massive disruption across the whole brain of information sharing. And that disruption at day 28 is in fact proportional again in each animal to the amount of pain behavior they exhibit. Okay, so at day 5 there is nothing, and day 28 again these two match with each other. So this is a very nice replication of the human data that I showed you in a much more experimentally sort of amenable sort of organism where we can be more aggressive to look at mechanisms. In the same data set, again, we do resting state functional imaging in these rats and look at the whole brain connectivity properties. At day 5, when they still have pain, at day 5, there is no reorganization in the brain, functionally. Day 28, all these lines are now significant. So there's a massive reorganization in the rat's brain, time-dependently, in relation to the peripheral injury. Okay. Now I'm going to show you some data. These are electrophysiology studies. Again, the details don't matter, but we are actually specifically looking at neurons in this region, in the nucleus accumbens, where the neurons are tagged genetically to be either red or green, depending on what type of receptor they have on top of them. So we can isolate specific receptor subtype neurons, look at their properties, and the accumbens dopamine D2 receptor-related neurons are the ones that change even at 5 days in their excitability. So here's an example of how many action potentials this neuron generates. It's decreased dramatically 5 days later. The anatomy of these neurons have changed. This is the sham control. How many branches do these neurons have? The dendrites, the extent of the dendrites shrink with 5 days, okay? And that's significant statistically. And their inputs, the amount of excitability that's coming on top of these neurons, their synaptic input is changing. Yes?
How are you defining addiction? How are you defining the changes?
I'm not defining addiction. What I'm saying is the addictive circuitry is involved, and the nociceptive system has engaged the addictive circuitry. So this is the circuitry that people on cocaine, on alcohol, on caffeine, addicted to caffeine, addicted to gambling, addicted to sex, they all show this circuitry's involvement.
Thank you. That was the answer to my question.
Okay. Anyway, so we see a whole slew of physiological and anatomical changes that happen to this addictive circuitry in the brain. Okay, and that's probably all I should say about it and move on. But we can manipulate these neurons. This is the beauty of modern neuroscience. I can, in fact, give these animals a virus that will infect specifically these dopamine D2 containing neurons, specifically in the shell of nucleus accumbens, and then turn these neurons on or off by an activator and look at what happens to behavior. So we have two different viruses that we have put in. One introduces excitatory channels on the neurons. The other one introduces inhibitory channels on the neuron. And this is the physiological evidence for them. And those in both decrease and increase pain behavior. So these neurons, the amount of excitability of these neurons controls how much pain behavior these animals show. Okay, so in our assessment, this is the first causal relationship for any neuron in the brain that's related to pain behavior in rats. Okay, so this is a causal relationship. We're not doing anything except making these neurons a little more excitable or a little less excitable, and then the behavior changes. So again, giving us some confidence that what we have shown in the human. Remember, so what is really also fun here is that we have taken the human data, gone back to animal models, and now identified a whole series of mechanistic processes in the animal model that's inspired from hypotheses derived from the human clinical sort of brain imaging studies. Now, in the humans, I was talking about the whole limbic brain. In fact, now we have similar virus manipulation data across multiple different sites. I was showing you the data for the nucleus accumbens. This is the data for the hippocampus. This is the data for the ventral tegmental area. And in a sense, this network is the mesolimbic system involved in addictive learning, emotional behavior, motivation, all of those things. Right? Okay, and this is, in fact, data from another lab, again, now using optogenetic methods, it doesn't matter, looking at medial prefrontal cortex and showing that in neuropathic animals, the medial prefrontal cortex now is again controlling pain behavior. Okay, coming back to my overall concept, we show brain anatomical and functional and structural properties. All of these are part of this predisposition model that I showed you, right? So there are massive brain properties that are risk factors, and the details of these risk factors remain to be studied. These are very crude studies. I mean, they're very preliminary data in a sense. But the interaction between these and the injury, whatever that injury may be, is this 1 + 2 = 3, which creates this transitional state, driven at least by this connectivity and other things as well. And all of that creates in a new brain state with its activity pattern different, information sharing pattern different, and brain anatomical properties and the shared anatomical properties in the brain. All of them are distorted. Now, I should emphasize that in the patients where the pain went away, the subjects that were recovering, we don't see any changes. So there is nothing happening that we can identify in the brain of the people who are recovering. The changes we are seeing are only for the people who are persisting. So the ones who are transitioning to chronic pain, the brain is massively changing. The ones who are not, they already have the machinery, in a sense, to get rid of that pain, and it goes away. Okay, here's another way that I want to show you the concept. I've argued from my first slide that nociception is always there, and it accesses both the cortex and the emotional brain. And the interaction between them may or may not give rise to pain perception. With a big injury, obviously, this information traffic becomes a lot larger. And some subjects have the proper machinery, I'm assuming, from the cortex to control the limbic circuitry, control their emotional responses to that injury, and recover passively. If, on the other hand, there's a lot of traffic between this emotional circuitry and the cortex that basically is amplifying the nociceptive information's relevance to the organism, then this system drives the cortex and reorganizes it into a new brain that's carved itself to become a chronic pain state. And the beauty is that we have very nice correspondences between the anatomy and the physiology of the human and the rat, so we can go back and forth. And we can generate data from both of them to complement each other to show what are the specific mechanisms underlying them. Okay, I'm almost done. I just want to summarize what I told you. Since brain characteristics are determining propensity for chronic pain has to be thought of as a neurological vulnerability. It's not a peripheral nociceptive vulnerability. Okay? Nociceptors are involved, but it's the interaction between them and the brain that creates the chronic pain state. Limbic circuitry are the primary risk factors. That is the emotional state of the organism that comes to that injury is a fundamental determining property. And that interaction between limbic and nociceptive systems reorganizes the cortex to a new chronic pain state. And also that, we see a beautiful set of at least correspondences between what's happening in the human brain and what's happening in a rat model. And again, my overall message is that the brain is, in fact, chronic pain is a state of the brain being addicted to nociception. Being addicted to nociception simply means that the relevance of that nociceptive input to the organism has changed. That organism values that nociceptive system very differently than that of healthy subjects. Okay, I'm almost done. And I want to thank all the students who have worked with me over many, many years. I do the talking, they actually do the work, and also all of the NIH funding that we have received over many, many years that has made these studies possible. So I'm going to stop there. Thank you. Excellent.
Okay, so we've got about 15 minutes for Q&A, and we've got a hand just a little bit down the row, and then we've got a second one a little further forward, and then I see a third one over here in the black. Okay, perfect.
Thank you for such a great talk. So with these relationships in the brain, since you're able to identify people who are at a higher risk for developing chronic pain, are there any studies that you know of or any people who are working on using that subgroup who you have identified these limbic pathways and systems and actually provide an intervention to stop them from going into that maintenance phase to turn it into more of an emotional experience instead of like a nociceptive, painful experience?
We just finished a study like that. So I did not emphasize much in my talk. This circuitry has massive dependence on dopamine. Okay, so and in fact we had animal data that shows that if we give humans dopamine plus NSAIDs, maybe that would stop the transition. And we've been doing this for the last 3 or 4 years, using our model to identify who is at risk and only treating the ones at risk. We just finished the study, and I can report to you that the results are completely flat. There is nothing in them. Okay, this was a massive undertaking, about 5 years of data collection, and we simply failed. Now, I don't know whether the treatment was not large enough. We were being very conservative. If we did not start early enough, there are many open questions, but we just finished the study based on our model, first predicting the risk factors, categorizing subjects into risky versus non-risky. We have the data and we are still looking to see whether we actually validated our model in this population. I don't know the answer, but yes, we're doing this now in many other studies as well.
Thank you very much. This was a very mind-enlarging talk for me. I'm kind of new to this, and to look at pain as a learning experience is interesting. You mentioned a little while ago about dendrites actually shrinking. I was told years ago, and it might be wrong, that dendrites actually grew in a chronic, it's like CRPS. Could you speak to that a little bit?
I don't know that data.
Okay.
If I would know that data if it existed.
Okay.
So I don't know where that information came from.
It was from a pain doctor many years ago. So
Well,
it's,
pain doctors cannot measure dendrites of neurons. So these are not easy experiments. They're hard to do. That was a person who misread something or another, I suspect.
it was about 20 years ago, so obviously a lot has changed.
Well, 20 years ago, the chances of that experiment being done is 0.
Thank you very much.
We have a gentleman in black over here. And then I've got a person in the back and then another person a little further forward.
Hi there. I'm still trying to work out how to answer this question, but it goes something like this. So we're looking at this as a neurological vulnerability, right? That we have predictors. And I don't know if it's been studied or if you would just make some guesses on this, but are there some associations with other comorbid conditions? Because you have some data on the anatomy and the functional brain, but for us clinicians, being able to associate it with something in their history would also be helpful.
Yeah, I would love to report to you all those other comorbid parameters. We have very little. So, for example, in these patients, we have obviously monitored their depression and anxiety throughout this whole time period. Of course, they all have higher anxiety or depression as they enter into the study. They all have pain. They're not very happy about having that pain, right? And in the patients where the pain goes away, those parameters essentially go back to normal population. They decrease. The patients who develop chronic pain, their anxiety and depression don't change. They're exactly the same as they started with. And again, it is not getting worse. It is staying at the same level that they started with. Just like their pain is staying at the same level, their pain is not getting worse. It's exactly the same as it was at the time of entry. And anxiety and depression show similar properties. Now, in this population that we have studied was a massive undertaking. We specifically took subjects who had no pain for at least a year before they entered into the study, who only had acute back pain and had no other comorbid pain conditions. Now, that population, I challenge you, how many of them you can see in your clinic. It's a very small number. But the idea was we want to have a single chronic pain condition, and that is not contaminated with all of this chatter about, again, pre-existing pain conditions, predispose them, and all of that. So we have tried to eliminate those parameters.
So then maybe a prospective study would be the next step in terms
This
of...
is a prospective
This is a
study.
prospective study. So they...
Sorry, I
but-
used longitudinal. I could just as well use prospective. It's the same design. The design is we take a group of subjects and follow them
Sure.
as they change.
But in this case, they were in pain when you collected the data. There was those
Yes.
requirements, as opposed to having no pain to start and
Right.
then following through time.
Now, ideally, we should do the second experiment, but I'm too old to undertake that experiment, mainly because you have to wait many, many years to be able to collect the data, right? Most people, most of the time, happily, don't have pain. So if I sit and wait until you develop pain, I could be waiting for a long time.
We have a question in the back.
Thank you. Thank you, and very interesting information, and thank you for your research. Is there a role for direct non-invasive stimulation of cortical neurons or gene therapy in reducing a chronic pain patient's neurological vulnerability?
You mean something like a TMS kind of procedure, transcranial magnetic stimulation? Is that the question?
Yes, transcranial. In an article I read, 2015, promoting neuroplastic changes, considering the top-down approach as well as the bottom-up approach in cortical neuroplasticity.
Yeah, I mean, obviously, in my prejudice is that should be more effective than the bottom-up approach, right? So top-down manipulation should be much better. I mean, and in that sense, we are now doing many studies, which I did not show you, with placebo. Can I just, at least for small periods of time, make a big effect by just giving them sugar pills? And the answer is yes. And it is, again, predictable who will respond and who will not respond to placebo based on their brain properties. So yeah, so these studies are definitely pushing us into the individualized level of pain, at least understanding. I'm not sure about treating. We still are not quite there. I mean, but it's coming. There are lots of targets, and now multiple pharma companies are also excited and they're developing drugs, novel drugs to target these systems. Whether they will work or not, I don't know. But at least we now have a mechanistic approach of where to go.
We have another question from a gentleman in a tan shirt, just a few rows further forward.
My question was related to what she had asked earlier, and looking back at the animal models that you did with the excitatory inhibitory changes with the viral infection.
Yeah.
How did you distinguish between non-pain pain behavior versus just lack of motivation to participate? I'm curious if there was a way to distinguish, and on the larger scheme of it, as she was saying, is this something worthwhile as a route to continue for treatment?
So we actually separate motivation from pain behavior, and in fact, in that manipulation, motivation is not changed at all in these animals. What we are measuring is something very simple. It's the extent of sensitivity of that injured paw to stimuli, and that sensitivity goes away or decreases. That's the only thing we see that changes. Nothing else changes in these animals. The other body responses, their motor abilities, their anxiety levels, all of those, anxiety-like behaviors, for example, we can measure them and they don't change. So it's a very specific, it's a very minor manipulation, but it's giving us a very specific behavioral outcome.
Thank you.
Mm-hmm.
There's another question here on the other side of the aisle.
Hi, thank you. I suppose my question relates to the population. So you followed them over a year, and I wasn't clear whether they had any interventions in that time, any treatment, or whether they just did their own thing. And
good question. Yeah. Let me answer the question. Yes. So my hypothesis when we started the study was that there is no serious interventions out there anyway, so they can go and do whatever they want to do.
So that kind of leads me into that.
Sorry, I'm a basic scientist. I can make these statements.
Yes.
And so we let them do whatever they want to do. Okay, and all we did is track them. And yes, the people who were so in fact 50% of the population did not report as doing anything to treat their pain. They were just living with that pain, and in a sense they were in this concept that, oh, next week my pain is going to go away. And so even 3 years later, there were patients who have had the pain continuously and had not sought for help or treatment. That's number one. And now, if in fact we take the data of the treatments, whatever treatment they were using, and use it in our model, it is a significant parameter by itself. So yes, and the only thing we could show is that early treatment was more effective than later treatment. But as soon as we put our brain parameters in, those treatment effects simply were washed away. So now you can think of that as a good result, in that the treatment is co-varying with some of these brain parameters. But these parameters are stronger, so that in the model that we generate, those parameters disappear.
And I guess I just wondered whether how you would talk to, say, patients who are looking at this and may take a message away that it's a fait accompli, that if I show this brain activation, I am destined to chronic pain? Is there any optimistic words that
No,
you might
no, that's
throw
exactly
in
the
that?
point. So in a sense, even the positive results with the treatment, even though it doesn't survive the model, it shows these are probabilistic events, right? This is not that, in a sense, properly controlling this circuitry at that transitional state would give us exactly the kind of window opportunity with which to stop the thing from happening in the ones who are more vulnerable and the ones who are not. So look, if you go back, think of this data from another viewpoint, what we have shown, we don't know exactly how to treat the ones who are vulnerable. But what we can do is not treat the ones who are not vulnerable. So I have cured 50% of the chronic back pain population by saying they don't need any treatment.
Do no harm.
Right?
Yeah. So there is hope.
Yeah.
Thank you.
There was a hand in the very back. Was that an internet question or no? This is the gentleman. Okay. Thank you. Thank you.
I was just wondering if you've done any work or are familiar with any work looking at, for example, like the long and short dopamine alleles in relation to this.
Say it again, I'm sorry.
I'm sorry. I was curious if you've done any work or looked at any work where they were looking at genetic variability in terms of this vulnerability, for example, long and short dopamine alleles, that sort of thing.
Yeah, I mean, we don't do much genetics. There's a whole group of pain geneticists working out there. But in my model, I don't, if you noticed, there was a gene in there that we had put in. But there was a very small genetic study. The concept, of course, some of these anatomical structural properties must be genetically predetermined. But exactly what genes those are, our whole pain and genetics field remains a real mess, and it still needs to grow up, and we need to, to end up finding reproducible genes and mapping them to the brain. All of that needs to be done. It has not been done.
We had another question right back there. I see you guys up here. I'm not sure if we'll get everybody.
I'll make this quick. So thank you for this talk. What I remember from last year's Pain Summit, that with researchers using rats, that there is a difference in, I guess, how female and male rats do perceive pain. And I noticed in both the Baliki and the Wren articles, they both use male rats in particular. Do you have any, I guess, future research, or have you already looked into how both the female and the male brain, or at least in the rats, interpret the pain?
Well, I mean, in fact, we've gotten some funding from NIH to look at our data specifically as gender differences. At least in these data sets, when we've looked at it, we cannot see anything different. Now, the rat data, you're right, we were using only males. That's just stupid tradition. And we've moved on, and in fact, we now do both equal representation for both genders, kind of thing, even in the rats. But the human data is 50-50 between male and female. So, and in that data set, we don't see anything jumping out. There must be something in there. We're still looking. I want to find where is the sex chronic pain interaction. We have not found it yet.
Thank you.
I hate to say it, but those are all the questions we have time for.